protein synthesis. It's the process in which our cells make protein. So, to do this, inside the cell it has 4 organelles. The first organelle is the nucleus. The nucleus houses the DNA and RNA. In the DNA, it has ATGC, adenine, thymine, guanine, and cytosine. In the RNA, we have AUGC, which is adenine, uracil, guanine, and cytosine. Inside the nucleus is where transcription is going to happen between these two, the DNA and the RNA. So the RNA is going to make a copy of the DNA, and it's going to come outside of the nucleus into the cytoplasm and become mRNA, which is the messenger RNA. And it's going to go down to the second organelle, which is a ribosome. And that's where translation is going to happen inside the ribosome. So, that is over here. We have different aspects that are going to go into the translation. So, we have our little tRNA, we have amino acids, codons that will become anticodons, and a polypeptide chain. So our little tRNA is going to come down with its part, and it's going to have an amino acid, and it's going to connect here with the codons, and then another one is going to come along shortly, and the amino acids are going to connect. When they connect, they're going to start to make this polypeptide chain, and then one tRNA is going to leave. And they're going to keep going with this process until they have a complete polypeptide chain. So then you can see in the cell we have our polypeptide chain, and that's going to go into the third organelle, which is the RER. It's the rough ER. And it's rough because of all of these tiny little ribosomes that are in it that are bringing their polypeptide chains to it. And inside the RER, the protein is going to be folded. And after it's folded, it's going to be transported in a vesicle. That is called transcytosis because it's still inside the cell. Then, it's going to be transported over to the Golgi apparatus or complex. And inside here, the protein will be packaged and addressed. And then it's going to leave here, becoming post-translation, and it's going to go back into another vesicle, and it's going to go outside of the cell, which is exocytosis. So, ending it off with DNA is going to transcribe into our mRNA, and then it's going to translate into protein, and that's protein synthesis.